Welcome to FYI, the For-Your Innovation podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit arc-invest.com.
ARC Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARC. All statements made regarding companies or securities are strictly beliefs and points of view held by ARC or podcast guests and are not endorsements or recommendations by ARC to buy, sell, or hold any security. Clients of ARC Investment Management may maintain positions in the securities discussed in this podcast.
Hello, welcome to FYI, the For Your Innovation podcast. I'm Brett Winton, ARC Invest's chief futurist. With me, I have Nemo Despot. And today we're talking about neuroscience. Nemo, before we get to, we have a great show for you today. We're talking to Dr. Jin Lee, who does basically modifies brains, measures how they respond to modifying, including like cleaving off little neurons, turning them on and off, and then is creating a functional kind of map of the brain that should feed into a next generation of diagnostics you can get in the clinic with an EEG. Let's talk about the diagnostic space generally, Nemo, and think about both in the neuroscience side and on the cancer side, and generally, like with these diagnostic tools that are becoming more powerful, how does that translate into actual medical advance for us?
Yeah, so I think what is important to hear, just to start with like a big picture thinking, and kind of have some engineering flair. So, if you can't diagnose problem or identify problem correctly, you can't solve it. And so that's why, even the drugs and treating is obviously important. If we are not treating the right things, or if we are not assessing how the treatment is doing, we are actually probably not making the benefits. So that's why diagnostics is crucial. And that's why we believe that in healthcare, diagnostics is actually the kind of comes first, because if we can actually diagnose, and which means identify problem correctly, and then assess, are we fixing it or not, we are solving already half of the problem. And so with that in mind, ARC, we had been really kind of bullish when it comes to the diagnostic space. But what is the problem like in biology and life, right? We come back to this kind of idea of biological complexity, right? The DNA of 3 billion nucleotides, which can make 100,000 RNAs, million proteins, 40 trillion cells, so we have multiple layers of different information that we need to basically able to read so we can actually diagnose what's happening. And so diagnostics had a really difficult task, given that all complexity of the biology, to actually develop the tools to read that biological information. When we think about diagnostics, there are kind of multiple different approaches, strategies in actually reading this biology information. So, there is something like blood-based diagnostics, where we are reading at the molecular level, is that the DNA, RNA, proteins, and this is Natera, Tempus. But there is also imaging-based diagnostics, which is also then trying to it's reading, assessing probably some molecules or just the structures, and based on that, it's actually giving us kind of some prediction. What is also key in diagnostics to think about, each diagnostic measurement, it's a snapshot in time. Biology happens over the time. So is that one snapshot enough, or we need to longitudinal measuring? So you can see this is a very complex issue that diagnostics is trying to tackle. And we are definitely entering in the new era now when it comes to diagnostics, where we were before.
Yeah, so I want to unpick a few things you said. If you think about at a very crude level, right, we have the recipe for our body is 3 billion letters long, right? And then that distills into functional chunks we call genes, of which there are 20,000 roughly. Okay, so that means your body produces 20,000 different proteins. So you could imagine if a disorder in one protein caused a single disease, like every disease was just like one protein being wrong, you would end up with 20,000 diseases. But like there's more than 20,000 proteins. Proteins can be folded in different ways. They're expressed in different ways in the cell. And also like changing one letter in a gene that causes the protein to do one thing versus changing in another letter should cause like different diseases. But our disease list is not actually 20,000 diseases long, not because there aren't 20,000 distinct diseases, but just because we're actually very crude and poor at diagnosing diseases. And that's, you can think of like two kind of areas we're improving. One is like, how well can we even measure this stuff? And you mentioned in the molecular space, the measurement tools are getting wildly more performant. Cost to sequence a genome going from $1,000 to, we think, $10 by the end of this decade. And then the cost to read kind of like the transcriptome, the stuff that's getting thrown off the genome and actually expressed into proteins, is also improving at a similar rate. And the cost to create even like functional bits that allow us to look for different parts of the genome. So Twist Biosciences produces kind of these templates we can use to design these tests. That's improving, that programmatic biology space is improving, we think, perhaps 1,000-fold over the course of the decade. So this upstream space is improving very rapidly in terms of just the precision of the measurement we can do. And, AI algorithms and the advance there can come in and make that data much more operationalizable. The challenge with biology is you have so much data, it's hard to cost effectively parse it. And so kind of the combination of the tool should allow us to diagnose the true underlying diseases to a much more fine-grained level than we could before.
Yes, and I think there's a few points to make here. So disease, as you were designing, disease happens at a very molecular level. Like one letter change can actually be lethal, right? But we are on the, when we talk about diseases and how we are observing them, we are at really macro level. Oh, it's a pain. I mean, like pain is such a crude measurement, right? Or some like other kind of, behavior or other kind of things. And so there is this discrepancy where the disease happens and what we can actually observe, right? And so we are kind of taking approaches from both ends to actually kind of, find where they meet. And, and the thing here is that, it's not only that there is 20,000 proteins, there is actually 1 million proteoforms. And then this change in a letter in this particular cell can be causing state A, but in cell B it can be kind of, different thing. So there is so much actually variability on the molecular side. And then we think about the phenotype side, our tools for measuring phenotypes are very, very, very crude. And so there is this disconnect, and that's what actually makes these things much more difficult to do.
Yeah. And so when you think of kind of like the ways in which this is we're improving at this, like a company that comes to mind for me is Tempus. They're taking molecular data mostly off their cancer diagnostic franchise and pairing it with the phenotypic data. So your clinical, like this is the data in your health record, the doctor's notes, to better understand and diagnose disease. And they're not just doing it in cancer, they're also doing it in neuropsych and in, what's their other area, in cardiology.
Yeah, so I think that Tempus is definitely what we call internally V2 diagnostics company. So this is new type of the diagnostics kind of company because they're doing exactly that, right? Biological information exists at multiple different layers. So, we have the sequence, structure, systems. We need to be measuring biology at all those three layers and combine them together. And this is what Tempus is trying to achieve. It's coming with genomics kind of testing, but then also kind of measuring the different phenotypic effects and trying to put them together. Where I see the future, it's going back to this kind of P4 medicine concept, predictive, preventive, personalized, participatory. And I think here personalized and participatory is everything important. I think we are going to come to understand, every disease is N of 1, basically. Each disease, every person's disease is a unique because that person is unique and there is a unique effects there. And when participatory is important that in addition to this kind of phenotypic stuff that we have now, EHR, EMR, EEGs, and other things that we can measure, and molecular stuff, there is something that's called deep phenotyping. So like Oura rings, Apple Watch, Apple earrings for hearing. Those things that can be recording a signal, but at a longitudinal level. So for the long time, could be also be important part of the puzzle actually in kind of bringing these three things together.
It's very clear to me, and I start out this conversation with Dr. Lee, kind of introducing the idea that we are still very crude in biology space that we really have, I would say, a limited amount of understanding of all of the layers of complexity that are going on. One way you could think about it is it's like just our understanding of the number of diseases that there are. And I think cancer is actually a space where we have made more progress in that area than in other areas of the body, because cancer is so clearly a genetically determined disease, and our ability to measure it genetically has improved markedly. And I remember in the early days of our work in the multiomic space, there was a patient that we encountered. He wrote a letter basically talking about how his cancer, under the insurer's framework, he was taking a drug that was only approved for breast cancer. Even though this was a male, he didn't have the cancer in his breast. But it was because he had the mutation that was, common with breast cancer mutation. And so the drug was the right drug to take, but the insurer was kind of like, he was having to fight with them to get the drug approved. And now we've moved much beyond that, where we understand that actually where that cancer originates, might be indicative of the likely mutation that's there, but it's not dispositive. As in, we need to measure not where it originates, but what's going on molecularly. And through most disease diagnosis, still doesn't happen, molecularly or really objectively. And so that's kind of the direction the industry is going.
Exactly. And the cancer, what we realize with cancer, that each cancer is a unique disease. And that's, the statement. Not only that, even when you have a primary tumor and then you have metastasis in the same patients, they're different diseases because at the molecular level, they are different. And so the only way for us, that's why we have to kind of really, and I think this is where we're going to end up, completely personalized disease. Every patient is N of one. Every disease is N of one. And that's how we can be approaching it. And so first step to there is, as you're saying, is diagnostics. And this is what we see, with Natera, Tempus, especially in MRD testing, how actually important and crucial this is. Even like Personalis, there was just the news today, actually last week, Personalis and their next personal test was able, for breast cancer, for 78 patients, to detect 50 months before the mammography, the recurrence of the breast cancer, and it was 100% correct. And so at the molecular level for us, basically being able to read the disease and diagnose it, that's the way. But we are still in the early days here, right? And I think this needs to be not only for cancer, but for many other things. I think you should be thinking about this longitudinal every monthly molecular testing that's going to be happening to all of us, and that there could be kind of reading things in advance.
Yeah, I think it's useful to unpack exactly how that minimum residual disease testing works for the likes of Natera and Personalis, where they talk about N of one. These companies are literally taking the tumor sample of the tumor. Minimum residual disease happens after you've hopefully fought off a tumor. You're in remission, but you need to track to know when it comes back, because then you need to be able to treat, and you want to treat as soon as possible because you don't want it to spread through your body. And so the way the test works is you take a section of the tumor and you go to Twist Biosciences and say, hey, can you make a test that can detect this tumor early if it comes back? And so then Twist produces kind of like the synthetic DNA that is going to look for that cancer. That's not everybody's cancer. It's your cancer alone. And it's important to have a very precise kind of like search function for that cancer, because you need to detect it in very, very low concentration in the blood when it's just beginning to come back. And so it literally is a cancer test for you for minimum residual disease. Right? Did I classify that correct?
You did correctly. And what is key here, why the Personalis test was actually so successful, because it's an extremely sensitive test. It can basically detect 10 to the minus 6, so if if the DNA molecule which is mutated is one of the million, it can detect it. And so this is the level of sensitivity that we need to basically have. And probably the question is, what if we go to the 10 to the minus 9? What can be, and so this is going back to the original kind of point, biological complexity, right? There's there's a huge this kind of complexity, and the tools are just getting there now. But with this, right, 15 months before the standard I'm sorry, I said mammography, but I was not wrong. It was radiographic imaging. But 15 months before that can actually detect recurrence. I mean, this is amazing, and so it's actually saving lives.
Yeah. Okay, so we've talked a lot about kind of like the molecular space and how like we're getting better at molecular tools. Our conversation with Dr. Lee was really on the kind of like imaging diagnostic side, I'd say, or, and even cruder EEG, where you're not even getting kind of like high definition imaging. How do you think these two intersect? Like, I will confess that I am she presents this idea that we're on the verge of solving brain disorders, or at least diagnosing brain disorders. I think of all of biology, the brain seems like the most complex and impenetrable organ. And I suspect that it's hard to actually molecularly tell what's going on in the brain, because you can't have a brain operating while doing kind of molecular dissection of it. And so you need to operate in the imaging space in some way. But the imaging tool still seems so incredibly crude. How do you think that this space advances, and how do you think about kind of where we're going to be with regards to our conversation with her?
Yeah. So few things there. I agree with you. I think we are still very early days. I think, brain is extremely complex. I do think that the imaging tools need to catch up and I think that we need some combination of the imaging and molecular together. How is it achieved? I'm not sure in a live system. Like, spatial transcriptomics is one way where you get imaging and molecular together, but like you can't do that on a brain. So, it's unclear to me what kind of way, how this is going to be done. But I think the imaging plus some molecular things together, it's what's going to actually bring us there. I mean, the other interesting thing to think about here is the approaches such as Neuralink and the others that they're taking. Is there a way like that when you have some input and you're inputting some signals and then recording and seeing what's happening to actually be recording kind of brain function? That could be, another approach, because the imaging at the moment in the brain is just basically measuring metabolic activity. I mean, energy consumed, and brain is the most kind of energy hungry part of our body, and everything the brain does is the kind of dependent on that, and so that's how we are measuring. But I still think it's very crude. So I feel that we need to kind of better resolution on the imaging side, and then combine something with the molecular level, or as I said, like Neuralink-like approaches somehow that you can kind of be stimulating and then reading it out.
Yeah. I mean, and also it's like even, I think it is interesting that, for example, Tempus got an AI algorithmic diagnostic approved on electrocardiogram, which is basically like effectively an imaging output. I mean, it's not imaging, but it's, an analog kind of output, non-molecular. And you could imagine, one of the things that she, mentioned is that like even doing an EEG, there's a limited number of people that are qualified to read out that EEG. And it used to be for genetic tests, there was a pinch point at the number of genetic counselors that then could relay the results of the genetic test to patients. AI is clearly going to kind of like cleave through those kinds of blockages, where it will be both better and more objective at understanding kind of the readout of the test, and then able to provide kind of a querying tool for patients or even doctors to say, okay, what does this mean for my patient in terms of what I should do going forward? And so Tempus got the first AI diagnostic approved in the ECG space, and they're going after kind of neuropsych. So you could imagine that maybe, electro EEGs reading off the brain also provide a mechanism by which they could objectively diagnose something, brain disorders that right now are crudely diagnosed via questionnaires with doctors.
So I have some insights here. So when I was finishing my PhD, I was doing a fellowship in a flagship pioneering, and with two buddies of mine that were there in a fellowship, we had a company idea, basically EEG, and then use AI and actually kind of be predicting everything in the brain. It turns out that the data you get from EEG is still very, very, very crude. I think that's, and very limited, and you record it for a certain amount of the time, and the data that existed there, so this is 2019, 2020, it was nothing. So how I think that this solution can be for a brain, you have something like EEG, but which is constantly on and it's recording. And then, analyzing that data and connecting, and something like that, I could be kind of seeing as effective. All these just snapshots, I'm not convinced with
Yes.
that.
You're saying you need longitudinal data in some way. And so then you need to be, it's not just the patient wears the thing in the clinic and you're like, oh, I got it. Instead, you need some solution where it's able to be worn over time or comfortably measured, such that it's as easy as having an Apple Watch on or something.
Exactly. And I think that kind of solution will kind of really unlock the brain. And we'll see who will get there.
At a high level, it's it's impossible to underemphasize how much data there is in the biological space relative to how much processing power we're throwing at it. So, you take large language models, Meta's Llama 3 is trained on 15 trillion tokens. So basically take all of the existing written language and compress it into an AI model, and you have 15 trillion tokens. Think of a token as basically like a word. If you were to take genetic information and compress it into tokens in a reasonable way, then the largest kind of genome sequencing project paired with clinical data to date, the UK Biobank, which has 500 or 600,000 patients, that would result to 400 trillion tokens. So, on the order of 30 times ish larger than what was used to train Meta's AI. And that's only on 500,000 patients, and that's only reading at the hereditary level. And so there is so much we have way too much data and not good enough tools to understand it in biology right now. And so you end up with and doctors don't know how to understand it. Research scientists barely do. The acceleration in AI, I think, will have the biggest impact on the biological space of any space because we're so early and so crude. And if you think about where that impact is going to hit first, where the most dramatic change is going to happen, I'd argue it's going to be in the diagnostic space because that comes upstream of all of the downstream things you can do. You can't effectively drug a disease if you don't know what that disease is, and you can't select the patients that actually have the disease. And a big problem in therapeutics is I'm taking a collection of patients that I think all have the same thing going on, but actually a subset of them have something else going on that, I'm not measuring finely enough to distinguish the two populations. And so as it turns out, my drug works extraordinarily well, but just for a subset of patients, I don't know how to distinguish between the two. And so that's to me, it seems like the direction that things are necessarily going to go. And it's a long journey to get from here to there.
Absolutely. And Brett, and I think still it's a lot of data when you compare to other areas, but then to the problem and complexity that we are trying to solve is so little data, right? It's 500,000 people, but there's 8 billion, people alive now. But what the people that were living before, right? All the human population, that was all different types of the, evolution was happening all the time and changing the things and trying out the things, right? So I think this is, as you said, biohealthcare is the ultimate big data science, right? And we are not even the early days, before the early days, pre-early days, I would say now. And just
think about the average patient experience of going into a doctor and it's like, I have an ache. That doctor isn't even reading your entire medical record. He doesn't have time to do that. It's kind of like you have to fill out a new questionnaire every time where it's like, this is my age, this is my sex, this is whether or not I've, had X number of things that you can check off if you remember you've been diagnosed with them. And so it really is the doctor is trying to approach the diagnostic journey from a nearly blank slate. It's clear that that's just because there is not enough doctor time to kind of successfully embed the data. And so kind of what Tempus is going after, in their vision is actually, let's have patients that are being diagnosed with a full understanding of their underlying medical record at the very least, paired with the new kind of molecular or imaging data that were coming out to end up with better diagnoses. And that should result in both better, treating patients appropriately given what they're going through, and then designing, cures, drugs, therapies that are effective for patients that previously were probably going, underdiagnosed.
Yeah, I mean, when you think about what is diagnostics and when doctors are doing diagnostics, right? I mean, that's basically, I would say, multi-level pattern recognition, and doctor, he has 30 minutes with you and he has to see all other patients, and he can't digest all the information needed. And we were not also having we still don't have all the information needed, right? And that's going to be a huge kind of pad. So kind of AI approach there, it's going to be solving the issues for sure, because it's going to be much better in kind of that pattern recognition than the doctors can do.
And right now, it's if you look at, people think of the health system as very inefficient. It's partly because of this. There's diseases aren't well diagnosed. If you look at the aggregate amount of health spend, the vast majority is on not diagnosing, not drugging, but just kind of managing, patients that are in distress downstream of that. So at a high level, our thesis on why the multi-omic space is important and interesting is because you're going to take that trillions of dollars in health spend that is inefficient on just managing people that are already downstream of the event that you needed to try to prevent, and you're going to shift it into the diagnostic side and the therapeutic side to provide, real functional help and even cures for the diseases that are plaguing the system. I'll leave off until we go with the interview with it. There was a company that read out data, just recently, Beam, on a disease, alpha trypsin deficiency, I think. It's a genetic disease. 100,000 people have this genotype. They have this mutation, but only 10 or 15% of them are actually diagnosed. So there's 90% of this population that is out there with this genetic mutation that is causing underlying health issues, shortness of breath, they get infections that they just don't know about. And the reason why is because there's, to date, hasn't been anything you could really do for them other than manage the output. You have shortness of breath. Let's try to figure, maybe it's asthma, we don't know. And then for people who are smokers, it's actually becomes very severe, and you can end up in, a lung clinic and have your life severely curtailed. So those are the ones that actually sometimes get diagnosed, not even always. And then the treatment they're offered is, extraordinarily expensive, $100,000 to $150,000 a year to just try to manage the condition, and it doesn't really extend their life in a meaningful way. So there is a candidate drug that could actually, you inject it, it's CRISPR based, and it's a base editing, flips the switch. It changes the mutation inside the body and effectively cures the disease. So, of course, commercially, you're first going to go after those patients who are already receiving this $100,000 to $150,000 a year treatment, because it's this is clearly better than that. But also it will mean that this 90% of patients that aren't being diagnosed, suddenly doctors will be like, hey, we need to run a blood test for this, because if you have this, we can fix it. And so, and that's at a level where it's we know what the disease is. There's a genetic test you could do to find it in patients. And it's not being done now because there's not necessarily, the patients that are sick with this are not so sick that you want to give them $100,000 worth of transfusions just to kind of manage the condition. But they are sick. And so there are many kind of diseases out there that people would consider rare diseases, but there's 100,000 people in the US that have it. And so I think disease is chronically underdiagnosed, and it's kind of this priming the pump where you understand the disease, you create the therapy using new tools that actually addresses and cures the disease that then causes a lot of people to be like, oh, do I have this? Is this why I've been having trouble walking up the stairs relative to people my age? And so it can really make human health better. I think that we are in an early stage of figuring out how to diagnose and address disease, and at an even earlier stage at doing so in the brain. You'll hear Dr. Jin Lee is really enthusiastic about our ability to improve our ability to diagnose disease in the brain, which we really are even cruder at. And I think you can look forward to having a better understanding of what's going on with your health, brain or otherwise, on a go-forward basis with these advances.
We are in the earlier days, even in understanding what is a disease and actually calling it, saying this is a disease and why it's actually happening, right? So, like, there is an additional layer of this kind of, for me, complexity is that, what is actually a disease, right? What is normal and what is homeostasis and what is a disease, what is adaptation, right? So there is all that kind of additional layer that comes into the play as well.
Yeah. Okay, well, enjoy the conversation, and thanks for joining FYI, the For Your Innovation podcast. Hi, welcome to FYI, the For Your Innovation podcast. I'm Brett Winton. With me, we have Nemo, and we have Dr. Jin Lee. Jin, you are a neuroscience researcher. You do lots of manipulation of, I assume, mouse brains and other creatures to understand how the brain works. I have a very high level impression of biology generally, that we are so early and crude in our understanding of biology, molecularly, what happens in the body, what happens in all the organ systems, what happens in the cells. Of all the technologies we look at, this is the place where we are on the outermost layer of the onion. And within biology, I feel like neuroscience is even earlier and cruder. One, do you agree? And then what is the state of play in terms of understanding how the brain works, and what does it mean for kind of human health, disease, and where we're going?
Thank you, Brett. I agree and disagree at the same time. We are at the outer layer of the onion, I agree. But I think we're about to penetrate through. There's a lot of findings that are coming together where I think we're literally at the beginning of a revolution.
Tell me more. Like, why is it an improvement in kind of like our tools and instruments? Is it like, what would I need to believe that that's true?
So let's start by talking about why we are not seeing the revolution yet. What was the obstacle so far? I think the biggest obstacle so far has been the fact that the field didn't manage to define the problem properly. Can we talk about what that problem might be?
Sure.
Yeah, so what do you guys think is the problem that we need to solve in order to address brain disorders?
Understand the brain at the systems level.
Yes, absolutely. To solve every problem, the key is to define the problem. If you cannot define the problem, you can't solve it. And the goal in brain disorder treatment is to restore brain function. So if you cannot define brain function, there's no way to restore it. There are a lot of dogmas in the field, to solve Alzheimer's disease, you need to remove the plaques, A-beta plaques. That may or may not be true, but removing the plaque is not the goal of brain disorder treatment. The goal of brain disorder treatment is to restore brain function. So when you do a clinical trial with Alzheimer's patients, you don't go like, oh, the plaques are removed, so it's success. If the memory is not restored, it's not successful. That's why we have lots of lack of success. And that has been the case largely, in my mind, because of the fact that we couldn't define the problem.
So how do we define the problem?
That's a great question. So to define the problem, you need to recognize that brain function restoration is key. But the reason why people haven't been focusing on that is the fact that it's very hard to define brain function. Do you know how to measure brain function? Can you think of a way that brain function is easily measured when you go to the hospital, when you feel like my brain's maybe not working so well?
MRI.
Well, I mean, typically in my experience, I like when I'm in conversation with someone, I can tell whether or not they're all there. So there's like a very like high level phenotypic, like I'm constantly assessing people's brain function.
How's mine doing?
That's true. That's true.
Yeah, but in terms of, knowing if you're having memory loss or if you're having like some sort of motor function dysfunction, there are ways to tell because we are constantly, yes, we are assessing brain function. I'm having a conversation with you. Oh, Brett's brain seems to be okay. But there has to be a more quantitative way to tell what the problem is. Just like making an analogy to a cell phone, if your cell phone's not working, you kind of tell like, okay, the calls are not going through or like some app is not loading. But you need to know a little bit beyond that. If, my memory is a little off, is my communication between this node and this node of the brain is, it should be at 10, but now it's at 8. There are ways that we have to be able to assess. The brain normally functions like this, but in someone who's a patient's case, okay, this communication pathway is weakened. That's why your memory is weaker. Or this communication pathway is too active, which is why you have tremors or like you have seizures, for example. And so these are the kind of things we need to be able to address. But is there a way to do that? Not really.
Not really.
Yeah, Nemo mentioned MRI, but currently MRI is only used in 2 different ways. One is to measure whether your brain looks okay, right? Looks matter, where if your brain is shrinked like this, okay, there must be something wrong. But that's very far from understanding if it's functioning properly. If you can... It is also occasionally used to assess function, but it's done in a research setting in a very limited fashion. And so I think it's fair to say that right now there is no clear way to assess brain function. And that is why nobody has managed to make that a goal of drug development or diagnosis.
Is that more kind of function? The tools that we have are just can't help us actually measure the function because they are, and then is that the function? There are many different functions. Is that the consequences? There are many different functions of the brain. How like it feels they're like layered problems here.
Yeah, that's the thing. The reason why we don't have proper tools to do that is because it is so hard. Right? The brain has like 100 billion neurons, and then there are many, many cells supporting it. On top of that, there's a skull that's protecting it. We barely have access to it. And so it's like, what does it mean to even understand? And so there has been lots of debates around it. Some people think that we need to understand every single neuron's connection and then map it all out to understand how it works. As an engineer, I don't really think so, but there have been lots of dogmas in the field where they were all competing with each other, and it has never been put together as a single platform of understanding where, okay, here's Nemo's brain function. Let's take a look.
Well, okay, so you've convinced me successfully that, yes, we still are on the outer layer of the onion. Are you going to convince me that like there's a way that we're going to penetrate through? So like if we don't understand brain function, one, it seems like that's upstream of a lot of stuff, as in even if you can like measure something doesn't mean you can actually manipulate the system to have it kind of restore. But I accept measuring as like a precondition. Are there breakthroughs in measurement, or is it just like, wow, this is a problem?
You touch on a very important point. So what does it mean to measure it properly? As a systems engineer, what it means to measure is you measure it to a point that you can decide how to manipulate it. Does that make sense? So that is my definition of what it means to understand brain function. If somebody has a tremor, and then you can show that which pathway is leading to that tremor, to me, what it means is that you can also manipulate that exact thing that's supposed to be 10 and is now 8. You restore that to 10. That is removing tremor. And so that is exactly the level of understanding we need. Fortunately, over the past 15 years in my lab, we've been trying to understand how some of these underlying mechanisms work. And as a result, we have managed to successfully make models of brain function that allows us to have that layer of understanding. So we literally, in the onion, we kind of penetrated through. That doesn't mean we understand the whole onion yet, but we found this tunnel down to the core. And so there is a level of understanding that we have in the animal models. But then, not only that, over the past 10 years, we have taken some of those principles and then applied it to making solutions for human brain function. And that is now starting to come into play where we have solutions we can apply in the clinic. And I've recently taken some of my friends and family who are suffering from very significant conditions, taken their data and then looked at it. And I now see that we can actually make sense of all of these patients' brain. And so I think I will have to show you that to convince you further. But
let me guess at kind of like the set of tools you're talking about. So, you can basically do optogenetic modification of neuronal activity, right? As in, I modify a neuron so it responds to light in some way, presumably in a mouse. I don't think we're doing that in human yet, but correct me if I'm wrong, right? And so then I can basically shine a flashlight on a neuron in a mouse's brain and it turns off or turns on. And maybe it's a cluster of neurons. I'm probably botching that. And so because of that, I basically can turn the switch on and off and be like, hey, the light's turning on and off, then therefore, but it's actually resulting in brain activity. I don't know, like food seeking behavior or something, or turning that off. And so then I can use that tool to then conclude, hey, these things do this and these things do that. Is that kind of like the, at least animal model?
That's the definition of how optogenetic studies are done. But as a systems engineer, I don't think that's enough. What optogenetics enables you to do is to have more precise switches, right? I like to use this example. So if you've never seen a phone before in your life and you just saw one for the first time, there's volume up and down button, right? And you're trying to figure out what that is. And if you have a crude finger where you press both buttons at the same time, it will be very hard to understand that these are volume buttons. And so, but if you are able to precisely press volume up and down separately, you can figure it out very quickly that it is like a volume up and down button. If you press it together, you can't. So that's the innovation of optogenetics. And so what optogenetics allows you to do is to press button precisely. But that's not enough. You have to press the buttons and see what the consequences of button pressing is. So when I press the volume up button, that volume up button connects to these, audio system and talks to them in this way, and then makes these algorithmic control, which ends up giving you the volume up in your speaker. You need to monitor and understand the algorithm underlying it. What we have done over the past 15 years in my lab is to use these very precise buttons and then also construct models of how it engages the system to create the volume up and down function. And so once you can do that, you can start to understand, this phone is not working very well. When I press the volume up button, it doesn't go up. That's good, but that's not enough. But then if you have the algorithmic understanding underlying that volume up button, then you can say oh, you know what? From this circuit board to this circuit board, there is a communication path that's broken. Let's find a way to enhance it. Then you can fix it.
How do you read those algorithms? How in your lab actually you read that underlying algorithm?
So we use various techniques. The first one we started using is functional MRI. And functional MRI is notoriously a tool where functional MRI is not accurate enough. There's blobs of activity. Neuroscientists, that's the way most of them think. That's one of the dogmas, for example, but once you have very precise control, the underlying information content goes up. And so we were able to take functional MRI tools to get a crude picture of, okay, these brain regions talk to each other in this way.
Maybe just for our audience, sorry to interrupt you, maybe if you could just kind of explain in plain language what is actually functional MRI. How should we be thinking about that?
Great. So functional MRI is an MRI imaging technique where you continuously image the brain while brain activity occurs, and the contrast is enabled by blood oxygenation level changes when you have activities in the brain. And so it has been a very useful tool for studying the brain because it allows you to, in live human brain, look at what goes on inside. However, there has been lots of, objection from many neuroscience community, that you need to know things at a cellular level. That one is too crude. We can't really make things be understood. And so it's a very useful tool. At the same time, there's been a lot of controversy around it. But one of the key ways, we have no tool. If we have a way to look at every single cell in Nemo and Brett's brain right now, I would, but there is no such thing. What we have to do is, with the physical limitations we have, put all the tools together and try to guess at what's going on. And functional MRI is one of the best tools to look at brain function non-invasively. And so that was the tool we started with.
And so just to make sure that I'm understanding this correctly, so basically then when the brain is doing the function, whatever is the function, we know it's metabolically active. So it's using the energy. And then with functional MRI, I'm measuring that kind of use of the energy basically. And then I do that longitudinally because I need biology happens over the time. So I can't do anything with a snapshot. So kind of combining these, so it's time plus energy consumption is kind of tell me the function. Tell me that there is some function happening, right?
Yeah, with spatial localization. Oh, here there's some activity happening. Here there's some other activity happening. And so that information we can collect, but how you interpret that is also a difficult task. And so what we've done in my lab is we use optogenetic control, which we cannot do in humans right now, but we did it in animals, use optogenetic control, combined with functional MRI, which is still, even in animals, there's no ways to look at every single cell. And so we use that with functional MRI in live animals to look at the overall brain network function. And once we did that, for the first time, we were able to understand how specific cells control brain circuit algorithms brain-wide. And once we had that context, we could also understand at a single cell spiking level how they interact with each other in certain contexts. And so that's what I mean by we have a nice penetration to the core of the onion. And then once we could so with that, what we are now able to do is to make brain function models at a brain-wide level with single cell spiking level to understand how certain brain functions work. And then on top of that, just as Brett mentioned earlier, with the level of understanding we have, we can make predictions. If I manipulate this part of the brain, I think this will happen. That actually happens. And so what it means to have a model that's useful is that it has to fit your data, and your data has to be precise enough to fit a model that is well-defined. And then once you make the model, you can understand how it works because you see the interaction. Okay, this is how it works. You have to at least be able to form a theory around how it works. And then you have to be able to make predictions on experiments that you've never done. And so we have now a model where that can be done. That's done in a mouse level, but we were not doing this in order to cure mice. And so this research has been going on for about 15 years. And in the past 10 years, we also then applied this to human data. And so for human translation, we decided to go with EEG measurements because functional MRI is a very difficult to deploy in in environments that are not equipped with like these, magnetically shielded rooms and stuff. And so since we have a very precise understanding of how things work, we could then try to get that information out of an even cruder data setting, like in EEG. And so we've implemented systems where we could take human data and then apply our findings from the mouse model. It's like taking pictures while knowing it's a face is different from taking a picture where you don't know what it is. So if I get like, if it's either a picture of Nemo or Brett, and then I take a picture of your eye, I'd be like, that's Brett. Or, this is Nemo, it's easier. So we have knowledge of how the algorithms work. And then with that, we could apply it to human EEG data. And we're finding lots of things that actually very precisely match what we found in animal models. And so not only have we penetrated to the core with the animal model, but I think we are also very close to penetrating the core of the human brain function.
So basically, as I understand it, at least the research breakthrough that you're presenting is the idea that using an EEG, which is pretty widely available, as in, you could go into a hospital, get it. You should be able to demonstrably assess certain kind of disorders, whereas before there was not something that would be parsable out of that data because we didn't understand basically the brain pattern that would result from the underlying disorder. And so then it was in the EEG data, but we just weren't able to read it.
It was always there. You couldn't figure out what it means because...
Can you give an example of like what is like a tangible example of something that you think within the, call it near future, you should be able to like be like, hey, statistically this person has this? Like, is there a specific like thing?
One area where we started to apply this in humans is in epilepsy, because of the fact that epilepsy is the only area of the neurology field. Any brain disorder, they don't really measure brain function in any other clinic. And in epilepsy clinics, because if you put EEG electrodes, when you have seizures, there's like activities that change. So even without very deep knowledge of how you of understanding how the brain works, you can tell like, there's an epileptic seizure, so there's something wrong with your brain. I think it's coming from this side of the brain, for example. But that actually doesn't portray the full picture of how it works. Oftentimes, it's very hard to even see what's happening. And so the level of human detection is very limited. And so we implemented the principles of what we found into understanding brain function where epileptic networks can be very clearly visualized, where, recently I've tried these on, friends and family when they were struggling with understanding what their brain condition was. They went to multiple clinics and they just got answers like, we don't know, and there's no way forward. But they have certain symptoms. They don't understand why that's the case. I asked for their clinical data and I ran it through our system, and we could tell exactly where the problems are arising. And I could even guess symptoms where like, does your family have this symptom? And so it is actually starting to show that, yes, these principles are working.
There's a paper this was a number of years ago. I'm sure I'd imagine you're familiar with it, where I don't know what it was. People were basically asking, could we use neuroscientist techniques to understand a computer? And so like some researchers went through and like basically like pulled apart a Donkey Kong machine or a virtual chip of a Donkey Kong and ablated certain transistors and kind of were like showed that, yeah, you could figure out what would break it, but you actually couldn't get to kind of like the function of what the chip was actually doing by operating at the transistor level and just like frying stuff. That's
exactly the point. That's what people have been doing. That's why they haven't figured it out. The approaches we're using is to figure out system function, where when system function context of everything, there's an A beta plaque there, but that's not the, that is, I don't want to say it's not important, but that's a variable in the system. you have to be able to deconstruct the whole system function level. Then everything is systematic and easy. When you take your phone to your like Apple Store or whatever, they don't struggle to figure out like, you have water there, so what does it mean later? It is where like you just open up, run system diagnostics. This is the part that's broken, and then you just replace that part or like, restore the software. That's because you understand the system at the whole system functional level. The approaches that have been used so far has been like these hypothesis testing and like, is plaque important or not? Of course it is important, but that's not the full story. And so there has been a lot of binary hypothesis testing, just like it's illustrated in the approach of that particular paper. And that's why we've been failing. But getting the full system level understanding, even for me doing that research, I ran into lots of objections. Where like, like academia and everywhere else, there are incentive structures built in, and it's not designed to look at it that way. Most of the people are familiar with hypothesis testing, and that's partly why the whole puzzle has never been put together.
In terms of your ability to like advance the research, if you like optogenetics is probably relatively recent. I mean, I don't know when that arose, but
it's relatively recent, 20 years. Yeah.
Okay, so optogenetics is a tool that we didn't have before. Then the fMRI side, are those tools becoming more precise or like higher resolution, or it's basically like this is as good as we can get it and you can't throw more physics at the problem?
So there are like tools like optogenetics, many other like precise neuromodulation tools that are coming on board, which is a tool set. It's a tool. There's also another set of tools, which is brain function measurements, fMRI, EEG, PET. There's lots of imaging tools that are also advancing where the resolution, precision, things are going up. But that's not enough. They're all tools. What we have to do is to put it together and then understand brain function. And then those tools have different meanings once you understand it. And what I think we now have is an understanding of brain function. We utilize those tools to understand it. At the same time, those tools can now be deployed to individually measure, okay, let's look at Nemo's brain. Let's see if it's working well.
But do you think we need more on the tool side development? Like when you think about what's available now, and I understand like in epilepsy and certain things, like you kind of, you understand the algorithm below, you understand the function. But as you said in the beginning, like, it's hard to define brain function because there are many functions, right? And so like, do we need more of these kind of brain reading tools development more, or like the ones that we have, we need more resolution? How you think about that?
That's a good question. Of course, if there are better tools, things will be better, but I think the limitation is not on the tool side anymore. The limitation is understanding what to do with it. And according to what we've tried so far, like in some examples, like let's say the tool's way here, and then, there are many layers of the best tool is at this capacity. We find that with our understanding tools that we are now using, the interpretation tools that we're now using, like we don't even need the best of the best imaging technology. We can get enough information out of even a lower layer of older tools, which makes it cheaper and easier to deploy in general.
How have you been advancing, call it the algorithm side, if I imagine I have the tool side and the algorithm side? Is that just kind of smart engineering and experimental design, or is there kind of like underlying advances in, how to interpret the data at scale?
So how to interpret the data is not an easy problem. Like if I give you that problem, Brett, try and understand it. It's very hard. How you do it has been debated by many scientists. Like, no, you have to do it like this. Yeah, you have to do it like this. But me in particular, we took a engineering approach of multi-scale analysis. And then what we have been able to accomplish is that I think we penetrated down to the core of the onion. And so with this level of understanding, we also then built clinical tools to do it. And then now we see that we can actually help patients. And so in this, Nemo's question was, is it the tool that's the limitation? No, it's not the tools. The tools are already there. It's been under development for many, many decades. And despite the fact that many approaches have taken, we need better and better and better tools. It's a very linear thinking. Better tools are helpful, but even good enough tools were around for quite some time. It's just that we didn't know what to do with it. Does that make sense?
Yeah, but when you say what to do, and how do you actually bring this to a clinic?
So what we have built is a software tool that allows you to take these conventional data and then use AI to automate a lot of different things that are very difficult to pick up by human eyes. And then based on our knowledge of what we need to pick out. And so AI automates those things. And then based on those automatically picked up events, then we also do brain circuit mapping based on the 15 years of research we did. Based on what we've learned, okay, this kind of activity interacting with that is important for disease. And so let's visualize it. And then we visualize it in state-of-the-art software tools. And then it automatically gives all the visual information that a physician needs in order to interpret it in a new way. Before, the physician would have to look at it and go, rub your eyes, go, I think there's something here. It's not a suited task for humans, in my opinion. It's a much more suited task for machines to take that into an interpretable form for humans, where what? Your network is communicating from here to here to here, and that this network seems broken. That's a much more interpretable form to humans than just looking at wiggly waves that's changing and going, what's going on there? It just doesn't work for us.
Yeah, at a high level in biology, it seems like the advance in kind of the diagnostic tooling and algorithm space is likely to expand our understanding of the number of diseases there are. And in neuroscience, it's like, if you read the DSM, like the diagnostic manual, they are such huge, broad, crude buckets that are basically like checklist style. Like they have 5 of these 7 things, therefore this is the disorder. And it's so clearly not actually, it's actually just like me talking to you and being like, oh, this person seems a little off. It's like at that level of crudeness. And so providing at least some objective mechanism by which we can tell what's going on and what's not going on in the brain is likely to unpack and uncover a lot of actual underlying diseases, or potentially even, clinically addressable diseases that before were all kind of overlapped into a set of readouts that we didn't really understand.
You're touching on very important points. I completely agree with you. And what I feel like, as you were speaking, I had this analogy in my head. It's like a blind person trying to categorize objects where they're touching it. These are all humans. These are all dogs or something like that. And so, there's going to be very significant limitation, which is why, like I said from the beginning, if you cannot define the problem, you cannot solve it. They cannot even define what exactly the disease is. But if you can see the brain function, so normal function, 3 of us all have different brains, but we're all humans. We have very expected range of behaviors that we're able to perform. That means certain functions like our motor control, our memory control, have specific normal functional range. Okay, we're all within that range where it all kind of seems like you guys are all good. And so that's the range. And if we can understand what that normal function is and specifically visualize it, solving brain disorders is very trivial after that. And
I'll take the over on trivial, but at least diagnosing brain disorders.
I mean, we do not think fixing a phone is impossible task. Phones are very complicated machines. We don't think of it as impossible. But if one thing goes wrong in the brain, it's literally debilitating, impossible task. because you can't figure out what's wrong. How do you fix it? So I don't want to say like fixing brain disorders is trivial, but compared to trying to go home with your eyes closed, that's going home is a very difficult task when your eyes are closed.
I think it's fair to say it would be a lot easier to try to address brain disorders if we actually understood what the underlying disorders
orders
were.
of magnitude easier. And
Sure.
what I mean to say by easy is it's not easy. It's where you can start to take systematic steps. No more guessing. I think I need to get rid of A beta plaques. Let's spend $1 billion and see what happens. You don't have to do that anymore because you can assess, okay, this is the next step you need to take. And when you take that step, the outcome is as expected. That's what I think of as the difference between like when you're doing scientific discovery and engineering. We engineers, when we decide to design like a phone or design something, we don't guess. We design it, and when you make it, it works as expected. That's because you understand the system. And when you can understand the brain system, designing therapeutics will not be like, we have no idea what's going to happen. Let's do a clinical trial and see what happens. It's more like, I think this is what's going to happen. Oh, it is what happened, but there's maybe some unexpected things here and there, but largely it is what I planned. And so that's what I mean by it'll be easier.
Yeah, I mean, within clinical trial space, you think of like even the failure in Alzheimer's trials is in some way, you're trying to like hit ultimately like a behavior modification. It's way downstream of whatever's gone on with the brain that set off that stuff. And you have a hard time actually measuring, like if your clinical endpoint is all the way at the phenotype, it's all the way at the, I'm talking to you and telling you if something is wrong. It's very hard to know if you're even making progress towards that clinical endpoint if you don't have a way to measure whether or not it's doing the thing in the brain that you need upstream of that clinical endpoint.
That's again, you're hitting on all the very important points. That's a very important point. So let's start with what you just said. When you're doing a clinical trial on, okay, let's get rid of A beta plaque, it's like a 12-year process. And then at the end of it, you know what? This person's memory didn't get back to normal. We fail. So not being able to see that memory recovery itself is a problem. But when you can see the brain function directly, you can much earlier tell that, okay, this drug is starting to restore the functional level. And so even if it fails, you fail earlier. And even if you succeed, you would know exactly how and quantitatively define it. On the other hand, also, not just can you assess it much easier, you can also design it according to your goals, right? So you see that in Alzheimer's, this is generally the network that seems to be off. Let's think about why this network is off. And then you can find hypothesis that makes very precise sense on, okay, like these nodes are generally not functioning. I see that gene expression of these molecules are in this place. That means, maybe I need to find a drug that hits this pathway. And then, you can, based on your visual information of the system architecture, you can even play a molecular level guessing game that's informed. And then you can see if that does it. And recently I was at a drug conference and I found out something that I didn't know before, which is that a single drug now costs $6 billion to make. That is not a sustainable
It's less, but it's still not sustainable. But just going back, to that, to your last point, and so this is something that we kind of think in ARC about. We really think that, today's healthcare is it's not a healthcare, it's a sick care. And kind of we need a paradigm shift to go from sick care to actual healthcare. And for that to happen, there's a concept called P4 medicine, preventive, predictive, participatory, personalized medicine. And so, that's how we see the future. And then I think for us as well is like why we think diagnostics is very important. Going back to your original point, with diagnostics, if you can actually define what's the problem really well, you can actually solve it. If I'm not defining the problem, I'm not actually solving it. And so how do you see this kind of, the stuff that you're doing, but how do you see this kind of transformation in general in kind of, in 3-5 years in the neuromedicine kind of taking a place, and what are the effects that you could be envisioning?
Yeah, so what you're talking about, the P4 medicine is exactly what this type of technology can bring about. So, I mean, we don't have to wait 3-5 years. Right now, what we can start to do is monitor everyone's brain function to assess where it is. So I envision that everybody should get regular health monitoring to see your brain function. And then once you have a brain disorder, you should get detailed assessment of what exactly the problem is. And then we can start to customize the treatments. The part we still have to make advances in is, now that we have this understanding of the brain, what we now need is the tool that can exactly intervene there. Those are still lacking because we didn't know what needed to be fixed. But now that we can clearly see what needs to be fixed, we need to then accelerate tools to address those things. And then once that is in place, what we can do is, we get regular health checkups. And then, if there's a problem with your brain, you continuously monitor it. And then based on what the monitoring results are, you just precisely apply it. What it means is you have a system where you can actively manage your brain health. It will no longer be something where, like, you know what, if I have Alzheimer's, if I have Parkinson's, if I have epilepsy, it's the end of the world. Like, that means you're no longer you. You're disabled. But what it means going forward could be that, just like when your phone is dropped in water and some circuit breaks, that's not the end of the world. You can assess it and fix it. And I think that's the future we can envision with the kind of technology that is coming online.
I think usually when your phone gets dropped in water and you have water infiltration of the motherboard, you just throw the phone away and have to start again. So hopefully we don't have to do that.
Yeah.
It does occur to me that, like,
That happened to me in Maldives.
Yes.
iPhone out.
Gone. Probably eaten by a fish.
Unfortunately, we can't throw away our brains. So we'll, like the reason why it's gone is because, the cost to replace it is less than the cost to repair it. But for our brain, we need to repair it. We're not going to replace it.
I mean, it does occur to me that I've never had an EEG done. And you could imagine, like, that kind of brain scan, if it becomes functional, like if it actually begins to generate, real data. And I don't even know if kids have ADHD, I don't think they stick them in EEG. I think it's just like kind of assessment. You could imagine that kind of like, hey, let's look at what's going on, becomes something that is a more standard way, even to diagnose. Like, I think with a lot of brain disorders in the neuropsych space, the diagnostic criteria are so loose and vary from provider to provider, and such that we don't even, like, is there a real uptick in ADHD, or is it just that it's like parents are bringing their kids in and asking to be diagnosed? We don't even know. And so to me, I'm hearing all this. I'm like, yes, we're still on the outer layer of the onion. This is very, very, very early. But
yeah, I mean, in terms of applying it, once we apply this technology, we are at the outer layer. What I'm saying is I think we have a little tunnel through to the core. What we have to do is, using that tunnel, start to peel all the layers. And exactly like you said, there has been, I personally think the disease is upticking for various reasons that I've discovered through research. But without quantitative assessment, there's very little we can do. But there are more things to do now. And one thing I want to point out also is the reason why you have never gotten an EEG exam and most people have not is not just because the results are crude. It's also that even if you want a crude exam, currently, there is shortage of staff who can read it. And so the accessibility is, like, even if you are an epilepsy patient and you urgently need to get an EEG exam, that's close to impossible. You have to wait in line for a very long time. But having an AI solution on top of everything else means that the accessibility is now fully opened, where what I'm advocating for is EEG for everyone.
And I can testify for ADHD, sorry, Brett. You get a questionnaire which you're supposed to fill out and few people around you, and that's how they diagnose it. Joke. It's a joke.
Dr. Jin Lee, I really appreciate you coming on. I'm looking for it. Maybe I should go get an EEG. I guess I'd need to...
Come to us and we'll get it for you.
Okay. I'm not sure I want to know. I think everything's working all right. We'll just stick with it. So really appreciate you coming on to the pod, and we look forward to a better future in brain health.
Can I add one more point?
Sure.
I wanted to say this before I go. Yes, we are making all of these solutions for brain disorders, but one point I would like to make is that all of these findings on brain algorithms are also going to enable next generation AI. Because AI right now, as much as it is exciting, has very severe limitations, which is that it uses unbelievable amount of energy for tasks that are relatively easy for human beings. And so us, as human beings, the human brain is designed to have very good energy efficiency. I believe after learning a lot about how the brain algorithm works, the brain's main design principle is energy efficiency. And so now that we're starting to understand brain algorithms, what it will also enable is next generation AI that's energy efficient. Because without energy efficiency, AI is unlikely to be useful the way we envision. And that's also another very exciting future that brain understanding of brain function will enable.
Yeah, I mean, our brains are wildly efficient relative to their computational ability. So being able to unpack and understand how that works so that we can apply it back into the chip space or the algorithm space certainly would kind of reflect back into the field of AI in a positive way. Okay, thank you so much for coming on the pod. And yes, everybody go and get an EEG if you can find somebody that can interpret it for you, or hire an algorithm to do it for you. Okay.
Yeah, we will just do it for you automatically.
Yeah. All right.
I'm coming. I want to know.
Thank you.
Thank you for listening to this episode of FYI, the For Your Innovation podcast. If you enjoyed this episode, please hit that like button and make sure to leave a comment down below letting us know what you thought. And if you haven't already, please make sure to subscribe to the show and follow us at ARK Invest on your favorite social platforms so you stay up to date whenever we drop a new episode.
ARK believes that the information presented is accurate and was obtained from sources that ARK believes to be reliable. However, ARK does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARK. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARK's current views and assumptions, and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.